And I'm going to introduce Sam Chavoshi. Sam is a nematologist, and he has a lab down in Tri-Cities. And before that, you were at WSU, weren't you? Or were you with USDA?
I work for WSU. Yes.
Okay. So anyway, when I have all of my tough nematode questions, I always go to Sam first. And I suspect that you guys, if you get into that field of soil science, you might do the same. Anyway, he's just a wealth of knowledge, and I'm happy to have him here today. I'm going to make him our presenter. Let's see here. Okay, so I think that makes Sam the co-host, and I'll turn the time over to him. And I think that if you click on sharing, you'll be able to share your screen with us.
Okay. Can you see my screen now? Oh. I can't hear you guys.
Oh, I think everyone's muted.
Okay.
But yeah, if you, I'm sure, Sam, if anyone has a question as we go along here, they can just butt in and ask it. Does that sound okay?
Yes, for sure.
Okay. All right, I'm going to put myself on mute, and you've got the floor.
So, okay, sounds good. Hi, everyone. My name is Sam Chavoshi, and I'm with Agnima, which is a plant pathology lab that is located in Pasco, Washington. And I'm gonna actually give you a little information about nematodes, but for sure, I'm not gonna go through the details of morphology, physiology, and those stuff. I just wanna give you a big picture of what are nematodes and what they do to the ag industry and why they are important. And also we will go with some of the most important ones especially in potatoes in our region, which might be more practical than going through the details of physiology and morphology, those things. I'm sure that you can find all those information in your textbook. So first of all, what are nematodes? It seems that you don't have my, let's see. Okay, here you go. The nematodes are non-segmented worms, and they are the most common animals on Earth. They categorize as animals. And it is estimated that something like 4 out of 5 animals are nematodes. And they already more than 15,000 known species are there, and it seems that there are much more than these numbers, but that means there are plenty of rooms for more studies on nematodes. And the majority of them are in actually in seas and oceans than on Earth, on ground actually. There are varying sizes. We have the majority of them as microscopic size, but there are very large size, like the ones that you can find as a parasite for blue whale that is 28 feet long. So And as one interesting thing is that we have nematode as a biology as a biology tool, as a model for drug discovery and also for studying on genes and biology. We call it C. elegans. It's very important especially in drug discovery. Nematodes are belong to phylum Nematoda, and the majority of them, maybe more than 90% of them, are microbial feeders. That means they feed on microbes and also on fungi. A, a very low portion of them are animal parasitic. Some of them are human parasitic. We do have insect parasitic. And what is important for us right now is the ones that categorize as plant parasitic ones. To give you an idea of the ones that we have as parasitic for humans, there are ascaris ones, there are pinworms, and you can find the information regarding the human parasitic nematodes especially on CDC with all information. So let's move on to the plant parasitic ones. And as interesting facts for nematodes, they have actually very simple morphology, no circulatory system, no respiratory system, and no skeleton. And they are highly capable of surviving in different environments. So all of these characters together make them very tough pests for agriculture. Based on the morphology, they have one particular part named stylet that is over here. And this stylet is actually the part that then they use for piercing to the cell walls and then suck up the nutrients. Based on sizes, and especially for the ones that we are talking about in agriculture, and if you imagine the tip of a pencil as 2 millimeters, so you can see pin nematode with very small size here. We have ring nematode with this size, root lesion nematode, very small size. And here are the size of nematodes like dagger nematodes. Dagger nematode is one of them, very large in size for plant parasitic ones. And you can find them as a parasite for all plants, especially for tree fruits and grapes. And they are capable of transmitting virus to the plants. So you can see the viral infection than a plant parasitic nematode infection first. Here is another picture of stylet, which is actually the part of the body that they use for feeding on plant. And here we have another picture when a dagger nematode pierce the stylet inside the root system and feeding on the roots. Okay, so the nematodes have different actually behavior for their feeding. So the feeding habits are categorized as endoparasitic, and that is the ones that their body stay outside of the plant, and they just pierce their stylet to the root system. Another category is actually the first one, endoparasitic, sorry about that. Endoparasitic are the one that penetrate inside the root system, and they stay there and they feed while they are inside the root system. The second category are ectoparasitic. They stay outside the root system, but they pierce their stylet inside the root and suck up the nutrients. And we have another category that in that semi-endoparasitic ones, they have a part of their body inside the roots and the rest of them outside. And these actually feeding habits are very important when we are dealing with nematodes, with plant parasitic nematodes. For example, for a nematode like lesion nematode, when they penetrate inside the root system, they open wound. And those gates, open gates, have the main access for other pathogens like verticillium, fusarium, and pythium, and they can have better access to the inside parts of the plants. Okay, to give you an idea of how they look like, you can see the root knot nematodes when they when they penetrate inside the root system here in this picture, and when they start feeding, they make some types of galls, and that's why we call them root knot nematodes. This gall is a combination of eggs at the end. After just a few weeks, you can see something like 400 eggs in one body of root knot nematodes. But they stay inside the root system, and they release the eggs outside. Here we do have lesion nematode. Lesion nematode is able to penetrate inside the root system. When they penetrate it inside, they open the wounds, and those ones can actually open the gates for other pathogens too. We do have cyst nematode. Cyst nematode have some types of actually semi-endoparasitic, and that means they have a part of their body inside the root system. But here, ring just use its stylet to suck up the nutrients. Also, stubby root nematodes is another one that stay outside of the root system. Okay, so let's talk about the importance of them in agriculture production and why they are plant parasitic ones. Mainly, they have quality and quantity losses for plant and for agricultural products. In potatoes, for example, they can make some yield reduction, but the main problem is the quality reduction. When they have some types uh, of bruising or problem uh, on flesh of the potatoes, this potato is not marketable, and the buyers or the processing companies easily reject this type of uh, potatoes. So with all those problems that they can make, they have a very big impact on agriculture. It is estimated that we have more than 100 billion loss for the damages that plants receive from uh, nematodes. And potato has a portion of 10 billion out of this one. And in that is worldwide view, and in United States, it is estimated something like $5 billion annually of crop loss. So as we mentioned, they have quality and quantity reduction. So they can make mechanical injury on the plants. They make actually changes in cells. That means they make cells to die, and also they change the growth of cells. When they make change in growth of cells, we see knots or galls. That one example is root knot nematode. They make physiological changes in their host, and uh, and one of the most important parts for nematode as a disease is disease complex. That means when they create opening for entry of other microorganisms, and that most of the time, that's the first signs of nematodes that you can see in a field. Mainly the foliar damage symptoms that you can see are very similar to water and nutrient stress. That is something like premature wilting, leaf yellowing, plant stunning, and irregular symptom patterns. But when you know your irrigation program, and when you know the water access for the plant, you can find out that there might be other problems that make this foliar damage. For example, here you can see some types of yellowing when you see when you look at the field from far away. Or here, the oval pattern of foliar symptoms. You may not see the yellowish here, but you can see smaller plants and some types of patches of problem in a field. Or here we can see the stunning in a field. This one is sugar beet field, and the pattern change based on the nematodes that they might present here. Also yellowing. Yellowing might be different from plants to plants, and based on your experience, you can find which plants has what types or pattern of yellowing. Again, stunning, and for crops like for carrots, the nematodes are like very dangerous pests, and the reason is carrot itself grow inside the soil and has nematode have access to the roots and to the carrot itself. So, and speaking of transmitting virus, here you can see the the virus symptoms in grapes than nematode actually problem. So here we have small bunches, poor fruit sets, and irregular ripening for a disease that coming from virus, not dagger nematode. But if you don't have dagger nematode, the disease or the virus is not spreading in your field.
Can I ask a question on that last one, Sam?
Yes.
So from a management perspective, is the producer best served to work eradicating the nematode problem or the virus problem or both?
Okay, for viral diseases, we have even more, or actually, I mean, we do have a very limited, you know, very limited system for curing the tree. Okay, so that means if a tree infected with virus, the first choice is to remove that tree. Okay, you can't work on curing that particular plant, right? So that means you need to prevent any viral diseases. Okay, uh, for fungal or bacterial, especially for fungal diseases, you might have more choices for managing the disease. But for viral diseases, we have only one, and that is preventing. For preventing disease, like this viral problem, you need to have clean plants, and those plants can infected only by nematodes to get the new viruses. So here, to manage the disease, the viral disease, you need to manage nematodes. Does it make sense? We do have some types of tests that you can check the nematodes if the nematodes already infected and harbor the virus or not. Okay, but when you do have the nematodes, the particular nematodes here, dagger nematode, if you do have it, that means the potential of transmitting virus is there. Okay. Okay, so for root damage symptoms, we do have gall roots, stunt roots, swollen root tips that comes from nematodes like, again, dagger nematode and stubby-root nematode, root lesions, and increase in lateral roots. Depends on the type of the plant that you have or the crops that you have in the field, you might see even either of those ones. For example, here we have galls that coming from root knot nematodes. And here we do have actually some small types of galls. Again, these are the cysts of cyst nematodes on roots. And for lesion nematode, you might see these type of lesions, but these brown actually parts of the roots are not exactly from of nematodes. They might be from nematodes and other pathogens that coming after nematodes starting to make infections, like Fusarium. That's very common for almost all plants, especially for potatoes and also for tree fruits. And here we have the root symptoms on potatoes again. They like, very large lesions on the surface of potatoes. And we have symptoms of nematodes. Those are the small dots here. I will show you more details about this particular one, but those are egg masses of nematodes inside the flesh of potatoes. Again, on potatoes and symptoms. Okay. The symptoms in different plants is different based on their, their system. The symptoms that you can see here are the symptoms of stem and bulb nematodes on onions. And that's one of the major pests for onions, especially for the exporters. When, stem and bulb nematode is one of the regulated pests for the majority of the export purposes. Depends on the country that you want to export the onions to. They might ask you for testing for this particular nematode, and they do have the same as a quarantine to make sure that stem nematode is not there. That's also important for processors. And again, for sugar beet, we do have sprangling of roots based on nematode infection. Another picture of Heterodora schachtii, that is one of the major problem for sugar beet. And this particular cyst nematode is present in Oregon and Washington. Okay, this picture can give you better idea of disease complex that we might have with nematodes. As you see in different treatments here, they use fungi. Here we do have fungi, Fusarium, I mean, and they are root knot nematodes. So root knot nematode and Fusarium together could make more problem for plants than either of them alone. We can see the control, Fusarium alone, and here we do have root knot nematode, one types of root knot nematode, that is Melodogine incognita. Here we do have Hapla and Javanica here. All three here make severe problem than Fusarium alone. Okay, so how to identify the problem? So you need to have a combination of observation on foliar symptoms, root symptoms, cropping history, very important, and laboratory analysis. Cropping history as kind of, let's say, data analysis for you is have a very important role, especially these days, for managing nematodes. Nematode might appear, in different numbers during the seasons. So when you have the history of a field, you can find them in a much better way than just looking at a single report. But you need to keep them as a part of your actually the history of that particular field. So you take the samples. Okay, let's talk about the identification first. For the identification has a very important role in managing the nematodes. Based on the identification, you actually you have a model of controlling, and based on the number of the nematodes that you have and the types of the nematode that you may find in a particular field. And also, as we talked before regarding exporting, when you are working on transporting plants from one location to another one, there might be some regulation involved for nematode and having the clean plants. So basically, the collection of samples for nematodes is very similar to the one that you do for nutrient purposes or fertility purposes. We mainly have 0 to 12 inches of taking the samples. But here, the difference between nematode samples and nutrient purposes samples are we are dealing with alive creatures. Nematodes are alive, and you need to keep them away from any hot and frozen, and you need to send them to the lab as soon as possible for analysis. And that's different from the way that you may handle samples for nutrient purposes.
Sam, I've got another question for you. You make the point that this sampling routine is similar to what we do for fertility analysis, and I would totally agree with that. The handling really isn't that different either with fertility analysis, but I've got to ask, how often do you get a sample in and find a nematode that's been cut in half by a soil probe?
Okay, so that's a very good question. When you take the samples for nematode, you need to take some, at least, based on the size of the field, that we recommend something like, for example, for potatoes, something like 5 acres for taking one samples. Okay, and you need to take something between 15 to 20 core samples, make a mix, and then have something like 1 pint of the soil as the final sample for sending to the field. Okay, so yes, you need to have the core samples, mix them, and based on that pattern that you have, you need to take some random sampling from 5 acres. Then the chance of catching the nematodes in that sample is high. But there are different ways of collecting samples. like grid sampling, that is a relatively new way of collecting samples. And through that, you can make a map of how they spread around the field. So, but otherwise, you do have them as patches of nematodes in a field. Yeah. Also, there are for plants um, like potatoes, you take basically you take two sets of samples, one from first foot, that is 0 to 12 inches, and second from 12 to 24 inches. And the reason is you have one season of growing potatoes, but the roots can go deep to even 3 to 4 feet down. So the chance of getting infected with the with nematodes is still high. And also for fumigation, that is one of the basic control system in our area is, they need to know if they are nematodes in lower part of the soil too. Okay, so let me see. Okay, here in this picture, you can see how they spread uh, in the soil, in the profile of the soil. The majority of them, the majority of the ones that we are dealing with for crops are in 0 to 12 inches, right? But they can go deep to 36 inches in this diagram that you can see. When you take samples from 0 to 12 and 12 to 24, you can cover the majority of them, and you can have the population that you are looking for to make the decision for controlling them. We do have root knot nematodes here. The pattern is different from root knot nematode than the root lesion nematode. And also you can see pin nematode has completely different. You can see the higher number in the lower part. But this pattern can actually change based on the plant too. For example, for mint, spearmint, when we take samples from 0 to 6 or 0 to 8, we may find a very high number of pin nematodes. So based on the plants, they might have different way of spreading in a profile. So this one is in a vineyard, Chardonnay vineyard. So that pattern actually formed based on the growth of the roots in a vineyard. So I'm going to show you this very short film, and this can show you how the nematodes look like in a in a nematode extraction. Okay, we have something like 250 cc of soil, something around 250 grams or half a pint, extract them, and the nematodes will look like this one. I have to say that this actually came from a healthy soil. And this number of the nematodes that you can see, for the majority of them, are beneficial nematodes, not the plant parasitic ones. That means they are the ones that feed on microbial and microbial part of the soil, and they can make the other nutrients available for the plants. So we categorize them as beneficial in a soil biology. But among them, we might have, plant parasitic ones. And the role of a laboratory is to detect the plant parasitic ones among those ones, okay, based on the morphology and or DNA analysis. Okay, for management, we do have the different ways, and for sure, you need to take or to have different methods at the time to be able to manage those nematodes. We do have the regulations. I brought regulations as the first one here because we do have nematodes in our area. We call them actually the reported nematodes. Those are the ones that are already detected and reported. But we do have some nematodes that we haven't actually found in our area. For example, potato cyst nematode. We don't have it in our area in Washington and Oregon. So it is still, it is, there are regulations that prevent them from coming to the area. Okay, we use that ones as quarantine pests, and we try to prevent coming those type of nematodes coming to the states. We have exclusion as much as you can actually, keep them away from the field. That's actually maybe the simplest and also the cheapest way of managing them. Sanitation comes with the tools, with the machinery, all those stuff that they use for different practices. Rotation is another way of managing nematodes. Different plants have different preference for nematodes. So you can use rotations for decreasing, not for elimination of nematodes, but for decreasing the number of nematodes in your field. Also, and maybe one of the most important one is plant resistance, which is the way that we need to work more on having the plant resistant as management method for nematodes. There are some types of even potatoes that are resistant to nematodes, and there are some other ones that working on them to bring the resistant genes to the more commercial types of potatoes. We have nematicides, and we do have fumigations. And as you might know, these last two are the ones that used the most, right? Fumigation and nematicides. As I mentioned, resistant might be, the future of our management methods. There are plenty of researchers on these one, and they are hoping for having different types of plants for resistance purposes. Right now, for soybean, resistant is the first choice for nematodes. And there are plenty of varieties that are resistant to soybean cyst nematode. Okay, we don't have this variety for potatoes, but there are plenty of programs working on this part too. I'm gonna actually talk a little bit about the potatoes and the ones that are very important in our area, Pacific Northwest. We have root knot nematodes, stubby root nematode, root lesion nematode, and stem nematode. Stem nematode is mostly important for onions, but I brought it here because of the importance with our rotations. So here are root knot nematodes. They are very small in size, something between 200 to 400 micron, and also we do have them as the main problem, nematode problem for potatoes. There are two types of root knot nematodes in our area. One of them is there are three types known in our area. Two of them are related to potatoes. Meladogine chitwoodi and M. hapla. Both infect root system and also the potatoes. The difference is based on the number of generation that they can have. As you see, Meladogine chitwoodi is the most important one. Can have 4 to 6 generations in one potato season. Okay, while the hapla can have 1 to 2 generations in one season. For that reason, we do have different damage threshold for these two root knot nematodes. If you check half a pint of soil and find one root knot nematode M. chitwoodi, okay, that means they already actually passed the damage threshold, right? Even one. But for hapla, you can go with something like 50, right? But for that reason, when you send your samples to a lab, they identify the species of this particular nematode too. Why? Because they do have different damage threshold value. How they look like in the on the plants, as you see here, we do have the chitwoodi that makes some bumps on potatoes. Hapla doesn't. You know, based on the shape, they are very similar, but they behave different actually on potatoes, and also they make you know, the severity of them are different. Here is a better picture. Again, for chitwoodi, root knot nematode, Meladogine chitwoodi, or Columbia root knot nematode, you can see the bumps on the surface of nematodes. And when you peel off the nematode, the potatoes, you can see the brownish part here. And those small, you know, browns that you can see are nematodes. What type of nematodes? The nematodes when they are in the egg mass stage. I can show you the egg mass. Here is an egg mass. Egg mass is a mass of eggs, right? So that means a nematode feed on potatoes like this, and then they have plenty of eggs inside their body. So nematode itself converts to an egg mass. a combination of eggs, something like 400 eggs in one egg mass. And when you smashed an egg mass, you can see these eggs. So you can calculate why one nematode per half a pint is too many, right? So with one generation, one of them can make 400 new actually eggs, right? And each egg have something like, or needs something like 4 to 6 weeks to go through 400 more, right? So again, another picture of how they look like. This one is another egg mass here, okay, on potatoes. And I'm going to show you this one. This one is the most important nematodes in Pacific Northwest. That is root knot nematode, Melodogyne chitwoodi, right? And they look like this. This is something like 200 times magnification under the microscope. But here you can see actually with higher magnification, maybe something like 400 or 500, okay, on your screen. Okay, let's move on. Okay, so we talked about egg masses, and I told you that these small spots here are the egg masses. They look like this, and then they contain something like this much of nematodes, of eggs. And also here we do have one single egg of nematode here. Okay, that is one of those ones, right? We dye them to be able to see them. And here are two pictures showing that potato tubers infected with root knot nematode. This particular sample, actually, and this particular potato storage rejected, okay, because of the nematodes. Okay, we had this particular sample last year here, and it was huge. The parts was huge. I mean, tons of potatoes rejected only because of this particular one. Okay, they have, mainly the processors have inspectors. They come to the storages, take the samples, and then either accept or reject it. They rejected their potatoes. Then they want to get another approval. They send it to our lab, and that was the the potatoes that we received, right? And here is the potatoes that or the nematodes that we found again. And in this particular sample, we had both chitwoodi and hapla on this nematode, on this potato. Okay. Is there a question here? So stubby root nematode is another important one in potato fields, and the importance of this nematode is they are capable of transmitting virus. tobacco rattle virus or TRV. They transmit virus to potatoes, and then we see potatoes like this. Let me show you this one. Okay, like this one, and these are not the damages of nematodes, but the damages of viruses. And so the TRV that cause corky ring spot disease is what we are going to prevent. So to prevent this one, we need to control stubby root nematodes. Another one again, root lesion nematode. This guy is capable of making damages on roots, but what is most important, again, is opening the wound, and then other diseases like verticillium can have access and make problem on the plants. You can see here when they penetrate inside the root system. Again, another picture here out of an apple roots. You can see the brownish part here. These brown ones could be because of the nematode, but mainly because of other pathogens. And we do have the complex disease here. So here you can see better actually picture of how they make problem with, particularly for this particular sample, we tested them and we found fusarium as the secondary to the nematodes. Again, we had this before, but this is stem and bulb nematodes. You can see this one as one of the most important problem in Pacific Northwest. This root is alfalfa roots, and they are one of the pests for crown and root system on alfalfa. And here is another picture. You can see, damages, very deep damages on these ones. Okay, and we do have the dagger nematodes. We talked about dagger nematode before. Here is a picture of those creatures. This nematode is the one that is capable of transmitting virus to tree fruits, grapes, and berries. They are huge, something like let's say 3 millimeter in size. So when you extract them from the samples, you can see them with your eyes. Okay, so I'm going to briefly talk about the actually extraction and identification of nematodes too, mainly. And as industry standard, we use microscopic examination. And there are DNA analysis available too, but the microscopic examination still is, the one that you can do with, let's say, less cost effective, okay, in compare with DNA. So mainly when we have the sample, the soil sample, what you need to do is based on the size of the nematodes, density of the nematode, and the gravity that you can make, you can use actually these three parameters to wash the samples, and then using the centrifugation and flotation system, you have the nematodes in very small amount of water. Then when you do have the nematode extracted, you can see them with the microscope, and based on their morphology, you can identify them. Okay, so if you do have this system, extracted nematodes, and extract DNA out of the nematodes, nucleic acid, then using the PCR system, you can detect the gene of the particular nematode and identify them based on their genotypes. So this system is more specific, and the sensitivity of the system is higher than a person that sitting by the microscope and checking them. However, this system is more actually expensive than checking them with the microscope and morphology system yet. But there are technologies that actually advancing, and we might have this one as as standard for the industry for identification of nematodes. Also, using this system, this qPCR or PCR system, you can identify if the nematodes are already harbor the virus or not. So you can find if the nematodes are or bring viruses to the field or not. They are just nematodes, and you need to take care of them and control them. Okay, with that, I thank you again, and I take your question.
That's awesome. Thanks, Sam. Do you guys have any questions for him before we let him go? I think we're probably all nematode experts now, right? At least on our way to being. Great. Well, yeah, thanks again, Sam. That was great. You guys will have this available. I'll put it up on Canvas, so if you want to go back and review some of the stuff, I suggest that. As far as housekeeping stuff goes, you've got a quiz that's due later today, so don't forget to do that. And then I have posted lectures 15 and 16 on the pages, so you'll be able to watch those at your leisure. We will meet. I'll get back to you on what we're going to do next week. I don't think we're going to have in-person lab. I think I'll probably have an assignment for you guys to do. So be watching for that. But I'll let you know before we get there. We'll probably do a lecture on Monday and then have an assignment on Tuesday. Wednesday is going to be review, and then we've got a midterm on Thursday next week. So good stuff coming up. Okay, any questions? Nope. All right. If you guys would give Sam a round of applause.
Thank you, Sam.
Thank you. Thank you so much. I try actually to give you some more practical things about nematodes, especially in our area, than more academic part of the nematodes. You can find actually more details about the academic part, in your textbooks. But those are the most important things in our area.
Yeah, I'm glad you talked about potatoes, because around here the potatoes are king. So yeah. That was good information. Thank you so much.
Feel free to reach out if you have any question. Yep.
Okay, goodbye, everyone. I'm going to shut it off.
Goodbye.